Welcome. Thanks for joining us. Welcome to today's presentation for the Envisioning Our Future for Children series. This is a celebration of the 100th birthday of the B.K. Ratchford Department of Pediatrics and the 90th birthday of the Cincinnati Children's Research Foundation. It also aligns with our strategic planning effort for the Cincinnati Children's Research Foundation. And we know today's speaker will help inform the direction of our work on behalf of the children and families that we serve. So welcome. This webinar will be recorded and will be archived for online viewing in the future. To claim CME credit, you must complete an evaluation that will be sent via email at the conclusion of the presentation, and this must be completed in 30 days. If you have any questions, and we welcome your questions related to the session, please enter them in the Q&A section of Zoom. You can access that at the bottom of your screen, and we will be sure to answer as many questions as we can, but please be aware we might not be able to get to all questions. If you have any technical difficulties, you can use the Q&A feature to contact our administrator. I'd like to welcome Dr. Jeff Mulkentine. He is one of our stellar researchers here at Cincinnati Children's. He's a Howard Hughes investigator and does work in the area of molecular cardiovascular biology, and who's going to be our moderator today and has collaborated and traveled with our speaker today. So welcome, Jeff.
Thank you, Tina. It's a real honor to be here today and have a longtime friend and colleague, Dr. Victor Dzau, as our guest speaker. I've known Victor for a number of years and have watched his science firsthand and really been in awe of not only his scientific achievements and contributions, but really a much greater platform for all the things he's done for global health. So as many of you know, Dr. Dzau is president of the National Academy of Medicine. Now here at Cincinnati Children's, we have a number of members of the National Academy, including our three previous chair of pediatrics, Tom Boat, Arnie Strauss, and Peggy Hostetter, and now our current chair of pediatrics, Tina Chang. We also have Dr. Jeff Whitsitt, Alan Jobe, Uma Cordigal, and just last year, Peter Margolis was inducted. So it's a real honor then to have Victor here with us. He's also vice chair of the National Research Council, and involvement of Victor in all the different national committees is really quite amazing. He's still a professor at the Duke University, where he was the past CEO and president of their entire health system. He was also a past chair of medicine at Harvard at the Brigham and Women's, and chairman of medicine as well at Stanford University, where he also led their cardiovascular group. Now, I've known Dr. Dzau for a long time because of his amazing contributions in cardiovascular disease research. He has pioneered a number of areas that have direct, tangible medical relevance, including the work on the renin-angiotensin system and its role in hypertension and its role in cardiovascular disease. And his findings have really led to various pharmacologic approaches to diminish renin-angiotensin system activity and the benefit that has on hypertrophic and dilated cardiomyopathies, but also in hypertension. And in fact, just this year, I think he's published one of his most seminal papers where they use CRISPR-Cas9 to delete the angiotensinogen gene from the liver of rats to basically cure their longstanding hypertension. And I think this has tremendous application to potentially thinking about lifelong treatments and a cure-like capacity for humans with standing hypertension. So in addition to all his many accolades scientifically and the more than 500 research papers, he continues to be involved in many different leadership level activities, including with the National Institutes of Health, where he's on their advisory committee. He's chair of the NIH Cardiovascular Disease Advisory Committee, and really more recently as the president of the National Academy of Medicine, especially these past 2 years during the pandemic. He has played a major leadership role for this country and global health, and his stances and position papers, and actually just his overall leadership and vision have been really quite remarkable for the course of the National Academy of Medicine. In fact, I've heard many say they've never had such a dynamic, but yet clear and on point beacon of light for the National Academy of Medicine as they've had with Victor. And it's really been quite amazing. Now, in addition to that, with the National Academy, he's involved and responsible for their global health risk framework and their positions and stances on that. their position and understanding of how the National Academy of Medicine is going to deal with human gene editing initiatives and how that would play out for the world, and many other countless challenges to health and medicine, both in the United States and globally. And his leadership, as I said, has been just tantamount to all that. Now, I'm just going to end with kind of an idea of his accolades and achievements at the level of prizes. Now, there's actually 2 full pages in his curriculum vitae of the various prizes. So I'm only going to select a couple to mention. And then we'll kind of get things moving here. So he's also, in addition to the member of the National Academy of Medicine, a member of the American Academy of Arts and Sciences, the European Academy of Sciences and Arts. He's won the Distinguished Prize from the American Heart Association. He's won the Ellis Island Medal of Honor, the Pulitzer Prize of European Academy of Sciences and Arts, the Henry Friesen International Prize, and he's got 11 doctorates, honorary doctorates. And I think the one thing he's most proud of is his single honorary doctorate from the University of Cincinnati, which was awarded in 2015. So it's really a great pleasure and honor to have Dr. Victor Dzau with us here today. And Victor, if I could ask you to go ahead and unmute yourself and share your screen, we'll be ready to roll.
Boy, Jeff, thank you. Tina, thank you for inviting me. But Jeff, you have taken about 10 minutes of my talk. And it was painful to listen to your exaggeration of what I've done, but I do appreciate it. As I always said, I wish my mother was here to listen. So she finally said, oh, Victor did something useful. But be that as it may, it's a great honor to be here. A great honor to celebrate with you your 100 years of the Retchford Department of Pediatrics at Cincinnati Children's Hospital Medical Center. So it's quite daunting when you look at your previous speakers, my protege, Jim Kim, my good friend, Don Berwick, and of course, David Altshuler, whom I know really well, and Aviv Regev. So I have, shall we say, this is a high bar for me to cross, and I'll do my best. But that being said, certainly I feel that what a great honor to speak at your venerable institutions. Can I have the first slide, please? So over the years, Cincinnati Children has made some major contributions to science and children's health. In your website, for example, you highlighted 4 medical breakthroughs. There's certainly a lot more. One is the Sabin oral polio vaccine, the first practical heart-lung machine, artificial surfactant. By the way, I hope Jeff Witz is in the audience. So I've got to know him well through the consortium of the stem cells consortium of NIH, and of course the rotavirus vaccine, really important. And of course, so many more years of discovery, science, too many to list. Certainly, I would say that people like Jeff Locatin and others certainly deserve also to be among this very, very venerable group. Next slide. So as I look at the issue that pediatric research and pediatric health is facing, Tina Chung actually published in 2016 a very important paper in pediatric research, which is a part of a strategic planning effort of the American Academy of Pediatrics. The committee conducted an open survey of board members of pediatric professional organizations and selected 7 great achievements. Look what they are: immunization, reducing sudden infant death, curing a common childhood cancer, ALL, saving premature infants by helping them breathe, ARDS, and surfactant. In fact, what came out from your institution, HIV transmission, mothers and infants, increased life expectancy for chronic disease. And indeed, if you think about this, cystic fibrosis, the average life expectancy was less than 25 years, 40 years ago. And of course, now it's more than 40 years old. And as we go forward, probably a lot more. Sickle cell disease, 14 years of average life expectancy 40 years ago, and now again over 40 years. And finally, saving lives with car seats and seat belts. So I would say that that's really quite amazing. And what about the next 40 years? And again, Tina led this article in which from the same study, look at what is going forward. More immunization, cancer immunotherapy, genomics discovery, big data and life course, interaction biology, physical, environmental, social environment to population health, quality improvement, and of course, reducing global poverty. I can't agree more. And importantly, Cincinnati children have contributed so many of the past successes and now are contributing to the future achievements. So what I want to talk about today is actually the foundations of all these great accomplishments, which of course is biomedical sciences and technology, and how they actually are helping us, but at the same time, producing more social issues that we must address as responsible citizens. Next slide. So, you know, it's interesting that 70 years ago, 1950, discovery of the DNA started this revolution in biomedical sciences, as adapted from Phil Sharp's writing, that there are, in fact, first in blue, the molecular biology revolution through discovery of DNA. In green, the genomic revolution that's also coupled with, of course, the Human Genome Project, the sequence of the human genome. And what he calls the third revolution, which I'll talk about, is the convergence revolution. That in fact, these things are coming together with all the other sciences that will make a huge difference in health and medicine. Next slide. So this is not a complete slide, but I would just simply that you all know the amount of biomedical breakthroughs in the last decade or so. I mean, it's really staggering. From genetic engineering, regenerative medicine, immunotherapy, synthetic biology, precision medicine, microbiome, and tissue engineering. All are amazing and hold great promises. Some already are achieving the promise of transforming health and medicine. Next slide. But you know, it's really quite amazing. Coincidentally, also 70 years ago, 1951, started the digital revolution with the first commercially available computer for simple data handling. But then this is followed, as you can see, by the first supercomputer, then by the ARPANET, which is the precursor of the internet, to launching of the World Wide Web in 1989, to ultimately where we are today, cloud computing, and of course, the iPhone, and so on and so forth. And next slide. And along with this, more recently are all the emerging technologies that is useful in health and medicine. Wearables, biosensors, telehealth, remote patient monitoring, robotics, exoskeletons, smart homes, autonomous vehicle, drones, robotics, automation. Not to forget big data and analytics and artificial intelligence. Next slide. So what can we expect going future? This is the convergence of scientific revolution. The revolution of biomedical science, digital science, but don't forget physical, chemical, engineering sciences. What you see here are three major reports, two by the national academies. The one on the left side, convergence, life science, physical science, engineering, and beyond. The second one on the top right, again, extending further. And the lower right is the MIT report to say the future of health is convergence. Now, many people say, well, we're already doing interdisciplinary research, but convergence is really a way to bring things together to problem solve. And it crosses all disciplines to form a comprehensive synthetic framework to tackle societal and scientific challenges. Many of these exist not only in one discipline, but in multiple fields. Next slide. So as we look towards the future, I would say the future is really bright. One certainly can think about, if you use the same 40-year projection that Tina talked about, that we're going to have more effective prediction and prevention, that we're going to better understand patterns of disease with massive amount of data, as been said in one of the predictors of the future of pediatric research, change the way care is delivered, right? Remote monitoring, virtual care, robotics, where care is delivered, and who cares delivered by, healthy at home, a seamless continuum of care with digitally connected, continuous monitoring provided in all settings, and of course, tremendous insight from big data, artificial intelligence, digital health, and of course, the breakthrough in biomedical sciences. Now, Chancellor Zuckerberg created this initiative, and their goal is to say by the end of the century, we should cure, prevent, or manage all diseases. Well, that's a tall order, but end of century is 80 years from now. Think back in 80 years before what we did have. So I think they may not be totally crazy. Certainly, if you use broadly, cure, prevent, or manage, I say manage for sure, prevent very possibly, and cure, well, we are optimistic. But it's still a tall order. And I think this is what I guess one way to say it is the brave new world of revolution in science, technology, and innovation. Next slide. But the other aspect of the brave new world is to understand that many of these technologies have tremendous implications for society. And many of these technologies cut across ethics, legal, equity, many other issues. So Salim Balabat, my specialist, and I wrote this article in Science Translates Medicine by saying, while we're looking at these advances, let's think about the health, but also societal implication of these advances. Next slide. So it is in this context that we thought about the brave new world. It was 1932 that Huxley wrote this, published this amazing book. I'm sure many of you read this book. And if I can see you virtually, I'm going to say a show of hands. And I think most people read it when they're in high school, whatever, about the brave new world. Now, what Huxley envisioned was that it's a dystopian world and that people are being genetically modified. And in fact, there's a social hierarchy which is dependent on intelligence. More so, he says, there will be amazing development in reproductive technology, in sleep learning, in psychological manipulation, and classical conditioning. Now think about what this book said, right? It is how many years ago? 90 years? And that's what I said about 80 years from now. In 90 years, where we are, think about this, we have actually many of those tools. Genetic modification, reproductive technology, psychological manipulation, conditioning, AI, you name it. So I would say that it is a brave new world. Technology and science is going to drive changes in this world, all for good, but yet we have to watch out for issues that may be unintended consequences, as Huxley thought about in the dystopian world. So as we think about scientists and clinicians and others, we have to think about how science has evolved over the years and what our responsibility is. The good news is over the years where science have really made great progress, there's been scientists who come forth to say, we need guardrails, we need safeguard, we need to do it, make sure that we are actually doing social good. Next slide. And the first example I think about is the Cinema Conference in 1975. This was a National Academy meeting which brought together 150 scientists to discuss the future of recombinant DNA, because in 1973, Cohn and Boyd and others were able to engineer a plasmid carrying genes for tetracycline resistance, insert it into E. coli, and the E. coli replicated and expressed the genes. This was the first cloning experiment, recombinant DNA. And Paul Berg led a group of scientists that wrote an open letter to the scientific community to say, we need to have a moratorium until we can assess how this hazards of such science can be addressed. And so, believe it or not, this was the first, to my knowledge, voluntary moratorium in the history of science. And therefore, in 75, the National Academies got them together at the urging of the scientists to talk about this. And they had a 4-day event. And you can see pictures. You can see Paul Berg over there. And there is Sidney Brenner. And you can see many of the people that David Baltimore and others. And ultimately, the scientists came out to say, we should proceed with this research, but we need strict guidelines. And that's what NIH Don Fredrickson did. Now imagine if we said, don't do it, right? Imagine if we say we don't do it. All the benefits of recombinant DNA we see today in research and therapeutics would have been, missed altogether. So I think this is a really good example where science needs to progress, but yet we need to come together being good people with good social conscience to say, how do we create the right safeguard? Next slide. Well, next slide, sorry. Next slide, yes. And then a few years later, there was Louis Brown, the test tube baby, in vitro fertilization. And she's now 43 years old. And of course, you can imagine the time people talk about Franken babies. And even Pope John Paul I warned the doctors, like sorcerer's apprentice, you may find yourself struggling to contain the consequences of your action. Well, 40 years later, 80 million babies have been born with this method. I'm not saying that the oversight of IVF is perfect, but certainly it's done a lot of good for those couples that are not able to reproduce themselves. Next slide. And then, of course, if you look at Dolly the sheep, cloning, here they took the nucleus of a somatic fully differentiated cell and put it into an egg cell, and then it was able to become an embryo that then got implanted, and out came Dolly the sheep. Immediately, the bioethics committee under Clinton and others, and almost now all governments in the world says, we don't think we should be cloning human beings. I say all these things in order to set the stage for my greater discussion today. Next slide, because we all know that the issue of science and technology in society continues to evolve. This book written by my good friend Juan Enrique, published this year, Right Wrong, talks about how society eventually, of course, decides whether advances go forward. But as advances go forward, our ideas of norms and ethics have also evolved and it's also shifted. And to a large extent, I'd like to spend a good amount of time talking about genome editing because it illustrates this issue extremely well. Next slide. So you all know this learned audience about human genome editing, the zinc finger nuclease, TALEN, and of course, CRISPR-Cas9, which the inventors won the Nobel Prize this past year. And everyone knows CRISPR uses a guide RNA that, in fact, targets a specific sequence or sequences on the genome, and then with Cas endonuclease, clips it in a molecular scissor. And in many ways, this is such a revolutionary technique. It's quite amazing. And the application, of course, even as the technology is coming out, people think about, wow, we can use it to really modify genes. So the application, of course, is either germline or somatic. Next slide. And next slide, thank you. And of course, somatic therapy, there's so many possibilities if you know the genetic defect. So beta thalassemia, sickle cell disease, even HIV, looking at the CR4 receptor and cancer. And of course, more recently, hereditary transthyretin amyloidosis that causes nerve disease, a product that's made in the liver, and Leber congenital amaurosis that in fact is the common cause of inherited childhood blindness. Next slide. So of course, it doesn't take a tremendous amount of imagination to say, let's go after some of these diseases that in fact are so central, they're genetic. And that of course is thalassemia and sickle cell. And you can see that many companies have really gone right into clinical trials using somatic therapy. Next slide. So I think an exciting report in January 2021, beginning of this year, report 2 patients from Oregon Health Science, you know what, I'm not quite sure what the organization is, I should know, but they look at thalassemia and sickle cell. They took autologous CD34 plus cells and they edited and modified the BCL11A enhancer, which represses gamma globin expression, which is responsible for fetal hemoglobin. So by doing so, as you can see on the right-hand side, after treatment over months, there's using the CTX001 infusion. You can see the blue color shows you that in the patient with, and the blue color, that the fetal hemoglobin has really been highly expressed, demonstrating, in fact, the success of the gene editing. And if you look at, that's the thalassemia. If you look on the right side, in the sickle cell, similar patient received this, and you can see a significant expression of the fetal hemoglobin along with some sickle cell, but this is significant improvement. And looking at the number of cells which carry the fetal hemoglobin expression, you can see it goes up to as high as 100% after infusion. The bottom line shows you, number one, the vaso-occlusive events in the sickle cell patient, the red dots, which you can see after infusion, 16 months, there's been no vaso-occlusive events. And also you can see in the blocks transfusion, and you can see that after 30 days in the beta thalassemia and 19 days after the sickle cell receiving this and transfusion, they're transfusion free. This is very impressive data. Next slide. Now, more recently, CRISPR Therapeutics, Vertex, this is through an announcement, of course, we have not seen the data, talked about 22 patients, 15 beta thalassemia, 7 sickle cell disease, and in fact, all of them are free from transfusion, and for the sickle patients, free of vaso-occlusive crisis. Very impressive. So we look forward to seeing more and more of this data emerging. Next slide. Now, as you think about this, where we've come along is this figure up there, from drug treatment to allogeneic transplant to ex vivo therapy, where you take the patient's individual progenitor cell stem cells and genetically modify them, to now what we hope that we're moving towards in vivo gene therapy. Because if one were to think about doing this broadly and equitably, the simpler, the better. And as you can see in the last slide, the idea of doing direct gene editing in autologous cells from a patient will circumvent the need for transplantation, and therefore you can actually achieve this. And as a matter of fact, we think about sickle cell patients and thalassemia, they're all over the world, and many of them live in under-resourced countries. So Gates Foundation NIH says, we're gonna really try to work hard to get this in vivo gene therapy working to make it affordable, easy to work in order to treat a lot of patients. I certainly wish them luck and believe this is the right thing to do. Next slide. Now, speaking of in vivo, already I'm aware of at least 2 successes in terms of proof of concept. One is, as I said earlier, the Oregon Health Sciences Center, they were able to look at Leber congenital amaurosis, where in collaboration with Editas Medicine Company and Allergen, they've been able to inject directly into the eye near photoreceptor cells. That's only showing that it's feasible. The second study, which had more data, looking at, in fact, what I talked about earlier about transthyretin amyloidosis, is to actually take 6 patients, and this is through Atelia, a company, And where they did is that they actually put it into liver because the TTR is made in the liver, just like what we did in angiotensinogen. And using nanoparticles, you can actually deliver CRISPR-Cas9 to liver, and they've been able to show a reduction in the level of TTR in circulation, about up to 90% reduction. So it's very exciting phase 1 study. Next slide. So with this in the background, I think one would imagine that this somatic therapy with gene editing would be really has tremendous potential. And I'm very excited about this. I think what is controversial, of course, is germline, particularly the idea of gene enhancement, right? High IQ, better looking. Now, the time of Olympics, being able to be one of the best athletes, don't we all wish it, right? Next slide. And that controversy, of course, is what triggered in 2015 the National Academy to begin to assemble people on this topic. Leading up to this, there was a small meeting in Napa Valley with Jennifer Doudna, David Baltimore, and others to talk about the potential usage of gene editing. And then upon encouragement by some of the people who were there, my members, such as Bob Horowitz, Keith Yamamoto, Mark Fishman, who talked to me about this, and Eric Lander talking to Ralph Ciceron, the former NAS president. Ralph and I got together and say, we should put together a summit. But, in 2015, we're not dealing with 1973. Research is done everywhere, not just in the US. So we decided to engage the Royal Society and Chinese Academy of Sciences and brought together people all to Washington, including social scientists, ethicists, to discuss about the potential aspects of gene editing. As a result of that meeting, we also put forward a report on human genome editing published in 2017 by NAM and NAS, chaired by Otto Cherrow and Richard Hines. And next slide. And this report says very clearly that basic research in somatic therapy can go forward because we have existing regulatory processes. But we need to limit any kind of research in clinical trials to treatment and prevention of disease or disability, not for an enhancement. Enhancement is a no-no. But when we talk about heritable gene editing, we said it's not ready, but it could be permitted after more research to meet existing risk-benefit and only compelling reasons. That was not a yes. This is more like you need to meet these criteria first. Next slide. What's also interesting, of course, is the public attitude towards this whole thing. Pew Research Center in 2014 looked at and then did again a survey in 2020, the public. And many people were quite cautious about the technology in editing. However, if this were to be used, you can see 15% says, yeah, you can do it for more intelligence-made baby, but 46% says, yes, I think we can use it for reduced risk of serious disease. 6 years later, you can see the public opinion has shifted. Now 66% can say, versus 46, you can use it for more serious disease and any condition, 60%, 57, any condition that occurred during the baby's lifetime. Still, people are very resistant to the idea of doing it for any other reason. Next slide. So this slide, in terms of green light, red light, I put together, all from George Daly, a concept. I think most people would say, yes, we should think about disease prevention, about these monogenic serious disease like Huntington, myotonic dystrophy, cystic fibrosis, sickle cell anemia, that we should not, in fact, think about enhancement, certainly at this time. And then there's a whole gray zone of diseases that I believe you can do somatically, that we should move forward, but not by germline. Next slide. Now, so all that is fine, and we were ready to organize the second summit. And the idea at the time was that it would be held in China. The Chinese Academy of Sciences well, should we say that they didn't like the many criticism that people talk about them, scientists moving too quickly. They actually dropped out. So the Royal Society and NAM, NAS still have the meeting. We enlisted the Academy of Science in Hong Kong, Lap Chi Choi, many of you know, a notable research genetics, and particularly in CF. And we had this meeting. And as I said, and I don't know how many of you read the code breaker about this whole area of gene editing written by Isaacson. And in fact, in there was accurate portrayal, including quotes from me of what happened at the night of before the genome editing meeting. Jiankyu He came and told us that he did it. And he made a public announcement that there's a birth of twins resulting in gene editing. And we were just shocked. The next day, when we didn't know whether he's going to show up or not, because he checked our hotel after a lot of threats, and he showed up. And of course, the place was bombarded with press. And I would say 1,000 people were there with many press. And of course, he was widely chastised for doing this, including by the Chinese, that he took HIV, which was not an unmet need. And there are many other effective alternatives. It was actually editing the sperm again. You know, that again, there are so many ways to actually, as you know, take care of that. There is reservation of fidelity of the editing. It's not clear about GMP, informed consent, lack transparency. And as you know, ultimately he was found for breaking the law by the Chinese government. And to my knowledge, he's still in jail. Next slide. But what that did say is that we're not clear enough in our recommendations. So Marshall McNutt, now the president of NAS, myself, and Cheng-Lei Bai, the president of Chinese Academy of Sciences, wrote the science editorial to say it's a wake-up call. Meanwhile, Eric Lander and Paul Berg, as well as Francis Collins, said we should have a moratorium on gene editing, heritable gene editing. Next slide. And that led to initiated two studies. The second study by national academies to go back and look at more carefully and greater depth with greater authority about what can or cannot be done at this time. And the WHO also setting up also a study. Next slide. So very quickly, because of time, I would say that our key message at the bottom is criteria for safe and effective heritable human genetics has not been met. So we should not be doing it. We don't know the technique, how good it is, on target and off target for human embryo use. And that on top, that no clinical use should be considered until clearly established is efficient and make precise changes without any undesirable changes in the human embryo. And that before any country decides to approve the use, there ought to be national, international mechanism to ensure that preclinical requirements have been met, and any clinical use, when it's ready, still have to be very cautious, restrict to a still small number of people with set criteria. Next slide. So specifically, this slide summarizes what the report says. First, you need to meet the translational bar. That is, if you're ready to use it, you have to say the preclinical research in cell animal models validate the methodology. Well, that's a minimum. If you look at drug development, that's toxicology and efficacy in animal models. You can't go straight to human. But second is, you also have to be sure that you study human embryos, maybe discarded embryos, to show that your technology have reliable on-target, no off-target, no mosaicism. Then when you're ready to do it, you have to take that patient's embryo, and if you're going to do it in vitro editing, it'll be important to look at biopsy, as we do now with PGD, a few cells to show that the edit is correct and there's no unintended changes. And then you need to show that it's developing according to milestones and unedited embryo before you can put it into implantation. And finally, of course, consent and important long-term follow-up, because we have no idea for sure that even when it's all done right, what are the long-term consequences. Now, if you meet all this, before you even use it initially for clinical use, can only be restricted to serious monogenic disease. And as you try to edit the gene, you have to edit to a common variant that's known not to cause disease. You don't want to cause more problems. You have to be sure that there are no embryos which are normal being subject to editing and transfer. And then finally, very few parents have to have almost no option or very few options. So actually the number of people meeting criteria is very small, and that's a toe in the water, if you will. Next slide. And finally, the governance is really important. That is, how do we decide whether countries should even permit to go forward? Well, it must be considered and informed by national consideration, and of course, greatly by the public. And social norms are going to be very important. Every culture, religion is different from country to country, and therefore it has to be considered in that context. And then you have the international oversight body, which allows to look at the science and whether it's ready or not, and to look at new classes of use. And importantly, in the case of the Chinese investigator, nobody knew who to report him to. So mechanism to raise concerns. Next slide. And quickly on the WHO, their report came out just now. Our report came out last summer, but they're more looking at across all the countries, including, of course, low and middle income countries. So their recommendation is that somatic therapy should go forward. And in terms of germline therapy, they believe that international collaboration is key. There has to be registry, a confidential way of reporting illegal and unethical research, and create a policy to take jurisdictions and domestic policies, among others. Next slide. Okay, so I've talked a lot about this. I think clearly the next step would be, what do we do with this? As I think in the chat, where's the teeth? I mean, there's a lot of work need to be done. The good news is that things have slowed down substantially, but you're quite right. There still could be rogue laboratories, etc., doing this. And so we are holding the 3rd International Summit, this time in London, hosted by UK Royal Society, to keep pushing in this forward direction. Next slide. Now, this is only, to me, a few examples of what we can see. But imagine the large number of technology, AI, and dual use research, many others. The science continues to emerge. So what do we do as citizens, as scientists? Next slide. I do believe that emerging technology and science have lots of implications and risk. One of which, of course, is the cost. The overall cost to care, the driving up the cost of care overall in healthcare, because certainly there are very few examples in which technology have taken down the cost rather than most of them will drive up the cost. Affordability, of course, is a critically important issue in that regard, mainly because of, you can imagine, socioeconomic challenge groups, access and equity, ethical challenge disruption of social norms, data ownership, sharing privacy and security, bias in digital technology, AI, biotechnology security, workforce, and training. Suffice to say that governance and oversight overall is outpaced by the rapidity of science and technology. Next slide. So these two slides will take a little more detail into some of these questions, and I won't take too much time discussing them. First, as I said, is how expensive will these technologies be? When you look at what's going on in some of the most recent technologies, that is treatment for extending several months of life, say in cancer, and treatment is $250,000 a year. I mean, it's unaffordable. So I think the real issue has to be, how affordable is it? And is it only available to rich or those who have insurance versus how is it affordable to low socioeconomic group? And how would it affect, of course, the cost care curve of healthcare? Inequity, how do you mitigate the biased distribution of the risk and benefits of emerging technology? And importantly, will the emerging technology exacerbate existing health disparities? Well, think about this. During COVID, we said, oh, this is great because virtual care, everybody's turned to virtual care. It's great news. A lot of people still have access to care, but we also know there's a digital divide. There are people who live in rural area, people who live in poor area, who actually have no Wi-Fi, no bandwidth. And I certainly, living in Durham, North Carolina, saw that. And we participate in finding ways to get, devices, et cetera, in this case for education. So this will further exacerbate existing disparities. How would it impact equity and social justice? This is a really important issue. We already talked about social norms, right? The whole issue about, do you actually now, these technologies available, are people under pressure to be looking better, stronger, because it's there, like Botox and others, and parents feel that children have to be more than perfect. And so how would society address these challenges? And what about doctor-patient relationship, right? Using AI in place of human, many issues. Next slide. I think, next slide, please. Yeah, and of course, everybody knows about data ownership, sharing, privacy, and security. We don't have a good governance structure, and we don't have a good way of data exchange. And in AI, it's very troubling because we all see AI as a great part of the future. But today, there are many studies that publish that AI algorithms based on data that do not represent the whole population often perform poorly for the underrepresented groups. Take criminal justice system. When they use the data to look at decisions about scoring, who's likely to have another offense. And in this case, it's greatly negative against the Black population and others. So they get more sentence, heavier sentence than less compared to white. Algorithms in healthcare, most of our data are collected around gender imbalance, male, white. And so there's data to show that chest x-rays and skin cancer detection are skewed because the database is not clear, but also many management decisions are really looked at, have bias against the Black people. And this ranges in heart failure, kidney failure, and many others. So I do think that these are really important issues, that how do we make sure the AI does not reflect bias inherent in many data sets. Synthetic biology, gain of function research, big debate right now around, of course, COVID-19. How do you actually look at and have a governance structure that really doesn't exist very well? And of course, training, and eventually with machine replace certain types of providers. Next slide. So in National Academy of Medicine, we have a 5-year strategy that started 2018. And we are now, of course, in year 3 and 4. And in strategy number 3, we look at science and how science is going to shape the future of health and medicine, of which one of the issues we talked about is to incorporate these advances, but to address social, ethical, workforce implications. Because we feel that we not only need to advance technology, we as academy and as the community need to address how do you mitigate the unintended consequences. As Marshawn and Willard wrote in here, emerging technologies involve a mix of applications, risk, benefits, uncertainties, stakeholders, public concerns. No single entity is able to fully govern any of this emerging technology. Just think about any technology. On one hand, you can look at science and the direct application. Then you have to think about, is it affordable? Well, is Medicare going to cover it? You can think about ethical issues, so legal issues. So we decided we're going to create a committee called Emerging Science, Technology, Innovation, CESTI, focused on health and medicine, to monitor and anticipate emerging technologies, to look at specific cases, to look at multiple factors that interact to shape technology, to analyze the risk and benefits, societal implications, and social disruptions, and to create ultimately a framework, a multi-sectoral framework to govern these technologies as they move forward. Next slide. We have really an all-star committee, next slide, that included Jennifer Doudna, who actually has, since Nobel Prize, has stepped down, but also have, next slide, please, people like Scott Gottlieb, the previous FDA commissioner, Rick Klausner, who's now in investment, Bob Kocher, who was the leader in the architect of the Affordable Care Act, who's now also investment, and people are scientists, George Daly, who is a social scientist, Arthur Chero, and Edison Hooney, who's actually been in industry and elsewhere, and people who are in the digital world, such as the head of Google Health, Qualcomm, and others. And we had a great team of people with the fellows and faculty from Hopkins working on this. Next slide. I would simply say the way we approach this is to do a case study. That is, we look at neurotechnology, telehealth, regenerative medicine, and genome editing to say, study the case, how many sectors does it interact, how does it impact many different ways. We then look at visioning. Where would we like in the future? And then we bring it together under a framework of ethical principles. And now we're working towards creating a cross-sectoral framework that we can say that you can actually begin to apply this thinking. Next slide. And quickly, you can see that what we are planning to do is to create an actionable guidance for governance emerging technologies. And such an actionable framework will need guiding principles, need analytic possibilities, and procedures. And what you see here are the elements that we're putting together. Won't have time to go over them, and they not put in order, just to say principles and values are important. We need to assess the impact on sectors, sectoral assessment. We need to do risk-benefit analysis. We look at scientific assessment, public engagement, looking at equity, diversity, and inclusion. And we need to give an overall assessment, but we also need to foresight to say what technology today looks like this, what could it look like in the future, understanding the forces that can influence it, and ultimately create a scorecard to say, or heat map, to look at emerging technology and create an assessment. That is shown on the upper right corner. And from the assessment, we need to monitor it as research going on and course correct if possible. Next slide. So I'm going to close by saying that the future is really bright if you go back to looking at all these possibilities I mentioned earlier. Next slide. But we, as scientists, clinicians, and socially conscious individuals, we know that advances opportunities comes without responsibilities. No longer can scientists and clinicians just do the science without considering social implications of their work. Frequently positive, but at times maybe not. So what is the social responsibility when it comes to science? The relationship of both positive and negative duties that we all have. Promote and protect the common good. That's what I talked about today. Now I'd like to show you my second last slide. Now in a paper that's going to come out in Lancet, I wrote a paper about where organizations like yours, next slide, academic health centers, children's medical center need to think along the way. We as an organization not only need to think about discovery and translation, but the ability to adopt and implement. Adopting implementation science, leading it to better healthcare, workforce training, but increasingly as the 7 important areas have pointed out, that as we think about how to take discovery to benefit the population, we need to think about social factors, cross-sector partnership, convergent science, social behavioral science, public health, and others. And finally, and importantly, as I talk about today, we have to think about society. You in Cincinnati, you are an anchor institution in your community. You hire people, you provide research, you provide care, you provide resources, and you need to think as a leader in the society, think about ethical, social issues, not only the work that you do, but also inclusion, equity, and I put down climate change, which was not, in fact, in the 7 areas. And I would say one more, mental health, really important area, because as you know, 1 in 4 people have mental health problems, and 3/4 of them begin in adolescence, going forward, anxiety, depression, schizophrenia. I'd like to add to those 7, at least 2 more. And you can see the integrating forces are data science, convergence, equity, and social good. So next slide and final slide. So what we're faced with is science is moving fast and the rest of the world is trying to catch up with us. So let's think about how to help in addressing this issue. Thank you very much indeed.
Okay, Victor, that was great, very inspirational. If you were in the room here, you'd hear everybody clapping loudly. We'll just pretend. So there are a few questions. Are you able to stay an extra couple minutes, Victor, or do you have?
No,
okay.
No, I'll stay.
Okay, so if everyone, we're going to probably take an extra maybe 7 or 8 minutes for some questions. There's some really good questions, and this was really thought-provoking. You did touch on the concept, and Amanda Zacharias asked about in 40 years from now, if you could put on your Huxley hat, how do you think climate change will have impacted global health?
Oh my, climate change already is causing a public health crisis. At the NAM, we are launching a major initiative on climate change. Because if you look at what climate change is doing, it's claiming over 20 million lives a year from malnutrition, poor water, extreme heat events, air pollution, you name it, globally. In the US, I think you can see increasingly the extreme events that happened with Katrina, with wildfires, and it's got many health effects. And of course, it's affecting the, shall we say, the vulnerable population, communities of color, even more than ever. I would say that if we don't stop it, 40 years from now, it will be the dominant reason why the world is collapsing, particularly when it relates to health.
I think that's a great prediction. And I bet a lot of us are worried about that prediction coming true, and that's unfortunate. So I'm going to keep the questions moving. Rafi Kopan asks, with this advent of genome editing technology, what if a rogue state, for example, in 20 years or 10 years, really decided to make genome edited super race, or whatever you would want to call it? I mean, what recourse does the world have to prevent or stop, or what are your thoughts on that?
Well, I think if there are some examples, such as nuclear treaties and others, of this magnitude, if this in fact is of that concern, because it's not a super race. They can make biosecurity weapons, bioweapons, you name it. So I would imagine this technology is still early. It's mainly sitting with the scientists in the biomedical community, but increasingly, that's why the WHO is on top of this. That's why others, I think eventually there has to be an agreement, a treaty of some sort, like the Nagoya and others, that look at bioweapons and also about the usage of this. In this case, you certainly, of course, the tools that you have will be sanctioned and many others.
Yeah, I love that answer. I think that's exactly what we'd have to do. And I know you're already thinking along those lines, and some of the things you guys have written. Mark Rothenberg asks, a lot of this technology, as we currently kind of see things unfolding with CRISPR-Cas9, is for monogenic disorders. But as we know, acquired disease or polygenic disease scenarios are increasingly a major part of health span and just lifespan in humans. So how do you envision these technologies really impacting, let's say, something as simple as allergies or autism or things that are?
Well, Jeff, you mentioned earlier about PCSK9 and our work on angiotensinogen. I think the problem with polygenic disease, of course, is you have many variants with small effects. So targeting one gene may or may not work unless you have a dominant one. However, you can know the common pathways. The common pathways that lead to disease can be targeted. And that's what you do in research, Jeff. That's what a lot of people do. And so in terms of angiotensinogen, it is a common pathway. And it's not all hypertension, but the majority of them look at ACE inhibitors, ALB. So targeting those could be useful. So I would say yes, somatic therapy, absolutely yes. As long as we can show safety, efficacy, and know the target.
I think that's a great answer. Tina Chang asks about getting back to your issue you kind of ended with, this idea of convergence and kind of the whole role that how this might impact pediatric institutions. And I guess what we wanted to do is pick your brain and ask you, what do you think we should be doing here at Cincinnati Children's to really position ourselves for the best scientific innovation for the future based on what you described?
Well, first of all, I put the last slide up, particularly for all of you and Tina, because I know she's responsible for the whole spectrum of research to clinical care. That increasingly, institutions like Cincinnati Children's has to think along that spectrum. So it used to be bench to bedside. Then it's bench to bedside to population. What we are advocating is bench to bedside to population to your society. And I think that's the right direction for any major organization to go. This paper will come out in Lancet, accepted, and will be coming out sometime in the fall. I think for Cincinnati, the convergence issue, Tina mentioned your institute or foundation, but more importantly, I think you are sitting also in a world-class university, and you're sitting around with many other institutions. I think that we shouldn't have to build everything, but reaching across the campus to bring in law and others would be highly helpful to create a convergence around problems. We had at National Academies had a 3-day meeting on convergence that the 3 academies co-sponsored. We have people who are provosts, presidents of universities, people like Steve Chu and others all came together to say, how do we change this? As you know, Jeff, the other one is funding agencies, because we're so siloed, right? If you do heart disease, you do neurologic disease, I think we need to think ways in which we can fund people to do convergent science.
Okay, I love that answer too. I mean, we are definitely, on the same page and thinking the same way, but hearing it from you, I think really helps us. I think we're probably going to let you go, Victor. This is great. We've already gone over time and taken, a lot of your valuable time, but this has been inspirational. We had this recorded, and I know this is going to be something that we're going to continue to go back to make sure we're on track. And I think,
if I can, I'd like to put a plug in for 2 things. Mental health. That's a big issue because, as you know, it starts off in the young, it's affecting the young. That's where research can be really impactful. And climate change.
I love it. Thank you again, Victor. And, keep up the good work. You really are, a strong voice of reason in an otherwise sea of confusion at times, and keeping the scope of science in healthcare scenario is so critical these days. And you've been a champion of that. And thank you for all your work in that area and all your advocacy for science and global health throughout the years.
Well, thank you for your great work in science, for Tina for inviting me, and for this great institution. I look forward to seeing you again in person sometime.
I hope so. All right, we're going to say goodbye to Victor. Thank you, everybody. Thank you, Victor.
Take care.